Hello. I welcome everybody to today's webinar about high-throughput genotoxicity assays for DNA damage and mode of action analysis. We will discuss how you can modernize your genotoxicity testing and in which points you can improve your test performance in the most beneficial way. I'm Dr. Jakob Bonse, Product Manager at Miltenyi Biotec, and I'm very proud to have Dr. Jeffrey Bemis speaking here today. He's the Director of the Clinical Studies at Litron Laboratories in Rochester in the state of New York. Thank you, Jeff, for sharing some of your expert knowledge on genotoxicity testing with us today. With Dr. Bemis, we will discuss simple and efficient assays for the DNA damage and multiple endpoint studies, and how high-throughput solutions improve your genotoxicity testing setups. Before, I will shortly introduce in the use of flow cytometry in genotoxicology and how our instruments, reagents, and software solutions will benefit your setup. With this said, I will hand over to the interesting part of Dr. Bemis. Replacement, reduction, and refinement. These are the well-known 3 R's of the concept first described by Russell and Birch, 1959, in their book, The Principles of Human Experimental Techniques. Round about 60 years later, these concepts are now the basis of many national and international laws, which regulate the proper use of in vivo experiments and laboratory animals. Even if we are still far from completely replacing necessary in vivo experiments, the younger past could show that use of appropriate flow cytometric-based in vivo assays is often capable of reducing animal experiments. Not only this, but rather more, they are also economically beneficial in terms of time, expenses, and convenience, especially compared to extensive microscopic-based analysis. To get a little bit more into detail, we'll have a look at varying regulatory background of chemical testing. The requirements differ depending on the region as well as the type of chemical under regulation. For example, in the EU, for cosmetics, in vivo experiments are prohibited, but instead different in vitro tests are required. In case of chemicals, biocides, veterinary drugs, or plant protection products, the regulatory requirements foresee to cover multiple endpoints by in vitro testing. Especially in case of biocides and industrial chemicals, a clearly negative result of this test impede further in vivo testing. The regulation for pharmaceuticals as well foresees in vitro testing in combination with in vivo testing. In the most of these cases, the choice of the right in vitro testing is crucial. Several OECD-approved flow cytometric-based tests can simplify your workflow and make the difference. Our MACSQuant analyzers bring the best qualification for high-throughput genotoxicity assays. For reliable and automated data acquisition, the MACSQuant Analyzer 10 is your trusted partner. With its up to 8 colors and the ability of our Express Mode packages, this very small-sized analyzer is more than capable of standardized and toxicological assays. The equipped violet, blue, and red laser are spatially separated and build the center of a very robust optical system. From startup to shutdown, the MACSQuant Analyzer 10 brings a broad offer of automated applications such as hands-free calibration, sample labeling, and compensation. Also very powerful is the volumetric cell count, which does not require any beats. This and more makes the MACSQuant 10 analyzer a very reliable and simple partner. Another key piece for your successful assays are the right reagents. In the past years, the subject raised that one main reason for the issue with reproducibility in data acquisition is based on inconsistent antibodies. In 2018, it was discussed that even antibodies derived from hybridoma are more frequently equipped with additional functional variable regions. To dispose this problem, we developed our own reaffinity recombinant antibodies. These antibodies are defined on the genetic sequence, which also ensures lot-to-lot consistency. The antigen binding regions are specified and validated for flow cytometry, and further, we use a universal IgG1 isotope backbone to simplify your experiment. Additionally, the Fc region is mutated to eliminate the background noise of the Fc receptor. This makes our reaffinity recombinant antibody a reliable and consistent choice. A not unimportant area in which you really can improve your analytic workflow is software applications. I would like to show you two beneficial applications which will simplify your in vitro analysis with high reproducibility. The first one are our express modes. To ensure automated and standardized data acquisition, we developed an algorithm which is optimized to gate your panel and thus the analysis fully automated. Express modes are sustainable even within high sample-to-sample variation. They will simplify your flow cytometry assays via predefined experiments, data acquisition, and analysis settings in a standardized way. The algorithm is developed and trained with a huge amount of data and adjusts the gating for each single data file individually. This ensures reproducible results. In this, we can say that standardized algorithm-based data analysis can reduce human error, creates reproducible results, provides standardized analysis based on mathematics, and simplifies your SOP-based processes. Everybody knows that if you measure the same experiment with two different devices, even if they are from the same type, you will get different results. Maybe you also already tried to manually normalize the differences in the photon multiplier voltages and now the struggle in this. Therefore, the second application, our smart gains, automatically calculates an intrinsic normalization and sets the gain for each device to 400. This ensures the exact same instrument setting from instrument to instrument and therefore an equivalent performance. This smart gains, like a lighthouse, consistently guides your instruments to reproducible and reliable data from test to test, independent of the user, on all instruments and all over the world. Standardization and robustness are the key points of our portfolio, consisting of optimized reagents, instruments, and software applications, which reach from research use only till the regulated environment. I hope you could find a beneficial solution also for your area. If you are interested or you have questions, please don't hold back and ask me after we listen to Dr. Bemis. Or you could write me an email to jakob@miltenyibiotec.de, or you take a look at our website, miltenyibiotec.com. Now I know we are all waiting for. I will hand over to Dr. Jeffrey Bemis, who will further discuss the high throughput applications on genotox testing. He especially is an expert in DNA damage testing and multiplex genotoxicity screening. Again, a very warm welcome, Jeff, and thank you very much for having you here.
Thanks, Jakob. I appreciate that introduction, and I also want to thank the audience for joining in with us today. So I'm going to start right out in our discussion of high-throughput genotoxicity assays with a quick little introduction about Litron, just to give you a perspective from where we're coming from. We've been in the genetic toxicology business for over 40 years and primarily focusing on the development of new and novel approaches, not only for preclinical product safety testing, which is where the bulk of our work is done, but in addition, we do some clinical research applications of these same endpoints as well. All of our assays are basically centered around flow cytometry, and that is going to be the theme of today's talk: high throughput instruments combined with high information content assays. So Jakob has given us a nice introduction to the instruments, and I'm going to focus for the rest of this presentation more on the assay side. The reason that we're doing this is we believe these higher information content assays can improve compound screening, have a nice complement to those regulatory required assays that Jakob was describing earlier. They can expand the range of information that you can get from the assays that you have to do, and then ultimately, again, as Jakob was describing, they do address multiple aspects of 3R's initiatives. So before we get too far into the assays, I want to define a few different terms that you'll be hearing me use throughout this presentation, just so that we're all on the same page. They might not be terms that you're familiar with if you're not a genetic toxicologist. So I'm going to start out with a couple of these definitions. The first is a clastogen. This is a classification of a type of genotoxic compound that results in DNA double-strand breaks. And you can see a picture of nucleus over there that has been experiencing exposure to a clastogen, in this case, a classic exposure of ionizing radiation. And this is very well known for causing these DNA double-strand breaks, a very cytotoxic lesion that we definitely need to be interested in if we're looking at genotoxicity. So clastogen is one type or a mode of action of genotoxicity. The next one that I'm going to be talking about is antigens. These are alterations in the segregation or the way that the cells handle chromosomes, primarily through division when they're lining up at the mitotic plate. As you can see here in this picture, we've got a nice division of chromosomes that are moving to either poles of the daughter cells, and that's the way it's supposed to be. All of these chromosomes are supposed to segregate equally. If you have alterations in that, if you had disruptions in the segregation, male segregation of those chromosomes, you can end up with angiogenicity, which is obviously a significant toxic effect going on in the cells. You can also get polyploidy, which is sort of an outcome where you have too many chromosomes in one and not enough chromosomes in another. I'll show you some examples of that later on. So we've got these two different classes of of genotoxic agents, clastogens and antigens. And finally, the other category that we kind of lump everything else into would be non-genotoxicants. Now, many of these may be just as cytotoxic as those clastogens or antigens, but they do that through other mechanisms. And in fact, they may ultimately damage DNA, but they're primarily doing it through secondary pathways. Some examples of that would be apoptosis or damage to lysosomes, et cetera. But it's not a direct type of DNA damage, such as we would see with either clastogens or antigens. I'm going to go over a couple of the current in vitro approaches. This kind of looks back at the slide that Jakob showed a little bit earlier and talking about the different batteries for regulatory genotoxicity testing. These are some of the specific tests that make up those batteries. And in fact, I'm talking specifically about the in vitro approaches here. So we're looking in this case at the Ames bacterial mutation assay. Some of you may be familiar with this as a classic assay that gets used all the time and is still a requirement for submission of compounds for regulatory approval. The COMET assay is another well-known assay that is, again, looking at DNA strand breaks, similar to what we were talking about before, that would be caused by clastogens. There are a couple of other ones that do get used every once in a while. That's the chromosomal aberration assay, which is, again, a chromosome damaging endpoint. There's some other assays, HPRT, mouse lymphoma assay. These are a little bit more specialized and not quite as commonly used as the final one that I'm going to be talking about, which is the in vitro micronucleus assay. And again, this is our indicator of chromosome damage that we're looking at. And I'm going to spend a significant portion of the rest of the talk addressing this with our in vitro micronucleus assay called in vitro microflow. So we're looking at micronucleus assessment in cell lines, and this is the assay that we have devised at Litron Laboratories. The way that this system works is we're using TK6 human lymphoblastoid cells primarily, and we've exposed them to our test article, the compound that we want to find out whether it's genotoxic or not. So within that mix of cells, we have some healthy cells, we have some micronucleated cells if there was a genotoxic insult that they experienced, we have some necrotic cells, and in some cases we may have some apoptotic cells. So we have this whole mix of cells after we've done our exposure, and then we start processing them. The first step is exposure to a nucleic acid dye A. This is cytox green in this case, or excuse me, this is ethidium monoazide that we use here. And what we're doing here is it's a red dye. I'm using that as the indicator on the right-hand side of this picture. And you're seeing that it is getting in and binding to the DNA of cells with compromised membranes. So those that are necrotic, those are apoptotic. Those that have experienced a significant amount of damage, not necessarily directly associated with genotoxicity, but this allows us to first pass, identify those cells that are dead or dying. Then the next dye that we use is Cytox green in this case, and we're adding that at the same time that we're adding a lysis reagent. So we're actually liberating the nuclei from those cells and looking at the nuclei, the micronuclei, and any other cell fragments that are in that cell population. This gets run through the flow cytometer, and I'm going to show you a couple of plots of that next. These aren't all the plots that we use for defining the different populations, but these are sort of the two more important ones. The one on the left-hand side has that Cytox green nucleic acid dye on the y-axis, and on the bottom, the x-axis, is that EMA red dye. What we're able to do is differentiate healthy cells from unhealthy cells. So the population that's kind of in that lower right, casting down in that region underneath the demarcation line there, those are considered dead or dying cells since they've taken up that EMA dye based on their compromised membranes. That allows us to get an indication of how much toxicity is happening and actually gate those cells out and only look at the healthy nuclei. So when we go to this final plot here, where we've kind of switched around the axes a little bit, I've moved that Cytox green dye to the x-axis in this case, and we're looking at forward scatter, or an indication essentially of size of the particle on the y-axis. That population, that blue population in the upper right-hand part are the healthy cells. So that's kind of, if you're familiar with flow cytometry, it might look a little bit like a cell cycle where you can see G1, G2M populations of cells there, and that's the healthy cell population. And then in the lower right-hand lower left-hand section, we're looking at the micronuclei. So those are much smaller and less dense than the intact nuclei, and therefore they are lower in forward scatter and lower in Cytox green fluorescence. That is where we actually enumerate the number of micronuclei that we are seeing in the cells. Now, that's not the only thing that we get from this. Because flow cytometry is a multiparametric system, we can get multiple types of different information out of that, not just looking at micronucleus formation. So in addition to getting those micronuclei and nuclei, they're a little bit more clearly defined here. You can see them labeled. We also get information on those main nuclei to look at things such as cell cycle, which can be significantly altered following exposure to a genotoxic agent. I'm showing you an example of that here, where you can see the gray profile is from a controlled cell type, and then the clearer dark outlined cell cycle profile is from a genotoxic agent causing a G2M block. So there's a significant change in the cell cycle that you can see there that's very characteristic of a genotoxic exposure. We're also adding in a bead, so we have a latex microparticle in there at a known concentration, and this allows us to look at the ratio between the number of nuclei and beads in each of the samples and give you an indication of cytotoxicity, so that when you compare the nuclei to bead ratio in the control samples versus the increasing concentrations of your test article, you can begin to see alterations in the number of nuclei, whereas the beads stay the same. And from that, you can determine the cytotoxicity based on indices such as relative survival, relative increased cell count, which is a very important part of the guidelines. If you're familiar with the assessment of genotoxicity for regulatory studies, you need to have a very good indication of cytotoxicity, because those two things kind of go in hand. Genotoxic compounds are by nature cytotoxic, and therefore you need to control that very well in order to dial in the assessment of genotoxicity rather than just looking at frank cytotoxicity. So we get all of this information, and that's a lot for a single cell, but we've increased the ability and the efficiency of the assay to give you not only lots of information about a single well, but lots of wells across a single study. And we're using that and taking advantage of the different types of analytical platforms that are out there and processing platforms. And the one I'm going to focus on today is over on the right-hand side. That's that Miltenyi MACSQuant 10 system that Jakob was just describing. The automated auto sampler on here that does robotic sampling of a 96-well plate is what allows us to do that high throughput walk-away kind of operation. So instead of historically where you used to have to sit in front of the flow cytometer and feed sample tubes in for every individual sample you needed to run, this machine does it for you and makes walk-away operations possible. This then allows you to significantly increase the types of studies that you're doing in terms of comprehensive dose response assessment. So in this case, we're looking at probably one of the most exhaustive executions of our assay in terms of looking at a single compound at 22 different concentrations per experiment. We're looking at a very close dose spacing, much finer than half dilutions. And based on the diagram that you're looking at here in the upper left-hand corner where we have the highest concentrations going down to the lower right-hand side, where you have the lowest concentrations, we're covering over 3 orders of magnitude of a concentration range, and we're looking in quadruplicates at each one of those experimental conditions. So we're getting a significant amount of information out of a single study, out of a single plate, to give you a very comprehensive assessment of what is going on in terms of that test article exposure. What this then allows you to do is get very fine detail in terms of dose response modeling and to apply things such as benchmark dose, which, if you're not familiar with it, is a type of point of departure metric that you can use to look at potency of different compounds. So what you do is basically come up with all the different dose response data for each of your test articles, and then you can rank them according to the different endpoints that you've looked at. So in this case, I'm looking on the x-axis at in vitro micronucleus frequency, and we've got a benchmark dose looking at a very specific change in the critical effect size, or the effect or the response of increasing micronucleus. And on the on the y-axis, we're looking in a comparison against gamma H2X staining, which is our DNA double-strand break. You would expect both of these to be fairly consistent with each other because double-strand breaks lead to micronucleus formation. And in fact, we're seeing a very nice correlation between those two endpoints in terms of the most potent compounds for either one of those endpoints being localized to the lower left-hand portion of the plot, and the least potent compounds being in the upper right-hand portion. So this is a very nice confirmation of the micronucleus endpoint that we're looking at in terms of ranking compounds based on their potency. So in terms of the conclusions for in vitro micronucleus and our microflow system, we're achieving chromosome damage assessment in vitro quite readily by flow cytometry. It's important to exclude the chromatin of those dead and dying cells because we don't want that to interfere with our assessment of true genotoxicity, as I showed you before. And we've determined that flow cytometry can provide more than just those micronucleus counts. We're getting information on cytotoxicity based on relative survival, relative increased cell counts. We can look at those cell cycle perturbations that can be caused by genotoxic agents. We're getting information on membrane integrity. And finally, a little bit of information on mode of action as well. I'm going to go into that in more detail in the latter part of this session. Finally, one of the things I wanted to discuss here really quickly is we've done a number of performance assessments. It's not just performance of the assay in Litron Laboratories. Since you guys are coming at this from potential users, what I wanted to relay here is a large multi-lab validation trial that we did with multiple different pharmaceutical companies, and where we sent them kits and they assessed them across a number of different test articles that we all looked at, and then we compared our results. And you can see excellent transferability, information on influence of apoptogens, as well as numerous other aspects of executing that assay across multiple different laboratories. That's in our citation from Bryce et al. In 2013. If you're interested in any of these papers, you should be able to find them quite readily online. Otherwise, you can request them from me. So now I'm going to move on to the second part of our session, where we're talking about our sort of additional assay where we're looking at multiplex genotoxicity screening in cell lines. So it's something that we're kind of positioning a little bit ahead of micronucleus testing, and this is using our in vitro MultiFlow system. The reason that we created this is there was a bit of a problem with those traditional in vitro genotoxicity tests that I've been describing to date, especially with those manual systems that are out there. There's a significant issue with throughput and scalability. So you really can't do high numbers of compounds if you needed to look at large libraries or do a lot of screening. It's difficult to approach those with those traditional systems. They also do have some issues with specificity in that there is an elevated false positive rate coming from those particular approaches, which obviously isn't something that you want to have to deal with if you can come up with better assays to replace them. And finally, mode of action information is kind of lacking in some of those systems in the fact that you can't get additional information beyond something is genotoxic or not. So our proposed solution was to identify a number of different biomarkers that we can use to discriminate not only genotoxic from non-genotoxic, but if it is a genotoxic compound, well, is it a clastogen, an antigen, or a non-genotoxicant? And we wanted to multiplex those biomarkers together into a simple add and read type assay. And what we devised is our MultiFlow system. And this is our one-step add and read type assay. So what you do in the upper left-hand portion of this diagram is you have the cells that have been exposed to your test article. You take the portion of those cells, you mix them with our working reagents coming from our kits, and all at once, all of the outer membranes are lysed and stained simultaneously. There are no washing steps. There are no secondary labeling steps. All of this occurs in the same well once you've mixed the reagents with the cells and you've got exposure to the different fluorescent antibodies. Again, just in the nuclei, like we were doing before, we've lysed the cells and we're isolating our analysis just to the nuclei of those cells. Specifically, we are using a nucleic acid stain to look at just those nuclei and identify them for further assessment of our flow cytometric analysis. We've included a antibody against anti-H2AX, so this is our DNA double strand break marker, as I was describing before. We also have an antibody in there against phosphohistone H3, which is our marker of mitotic cells. And then finally, the one you may be most familiar with is an antibody against P53, which is a marker of genotoxic stress. Specifically in this case, we're kind of looking at a nuclear translocation mechanism because, again, P53 is found in the cytoplasm, but since we've lysed the cells and we're only looking at the nuclei, we're essentially isolating this to all those P53 molecules that have migrated to the nucleus, and therefore that's where we're looking at our indicator of genotoxic stress. Similar to our previous assay, we're looking at including a counting bead in there as well, so we can get indications of cytotoxicity from the nuclei to bead ratio, similar to what we were doing in our previous assay. During development of this assay, we looked at a training set of compounds. We're looking at over 84 different compounds that were taken from this ECVAM list that were well established and characterized in terms of their angiogenic, clastogenic, or non-genotoxic potential. We're doing this again in TK6 cells, treating them in 96-well plates. We're looking at a solvent, usually in DMSO, top concentration of 1 millimolar. We're doing up to 20 different closely spaced concentrations using a square root 2 dilution scheme in single wells this time. There are concurrent solvent and positive controls on the plate. We do a 24-hour continuous exposure, but we can actually go in and sample them at different time points. Since this is a suspension cell line, we're taking samples at 4 hours and 24 hours across the 96-well plate, across the 20 different closely spaced concentrations. This gives us an awful lot of information about the test articles that we're looking at. Some of the examples of that information, we're looking at a prototypical clastogen here. This is MNNG, and on the left-hand side, we're looking at our indicator of DNA double strand breaks. That's the gamma H2AX marker. You can see at both 4 and 24 hours, we're seeing significant increases in that indicator of DNA double strand breaks because this is a clastogen and it is causing DNA breakage. The next panel over, the PH3, phosphohistone H3, is actually showing a reduction, and this is due to a mitotic block, which is characteristic of cells that are undergoing this clastogenic type of activity, and therefore this is characteristic of a clastogen to see reductions in phosphohistone H3 labeling. We see increases at both 4 and 24 hours with P53, our indicator of genotoxic stress, and finally our final indicator, polyploidy, we don't see any change in that particular endpoint because this is a clastogen. When we switch over to an antigen, we're seeing a very different set of responses here. So with the gamma H2X, we essentially see no changes throughout either 4 or 24 hours of exposure, but in this case, the phosphohistone H3 marker is significantly elevated, both at 4 hours and 24 hours. Over at P53, we see an increase in 24 hours, but we do not see a significant increase at 4 hours with most antigens. So again, that's a little bit different from the clastogens. And then finally, at 24 hours, we see a nice increase, a very robust increase in polyploidy, a very well-known characteristic of antigen exposure. Now, when you think about all of those characteristics that I've just been describing across all of those different wells, across multiple, say, 96-well plates that you've run in a day, this really isn't the most efficient way to look at data and to start getting information back out of your test articles and your studies. So what we really needed to do was think about different ways of analyzing the data and to move from what some people have called eyeballometrics, which we were just kind of doing on that previous slide, and just looking at the data, and switch to something that's more quantitative, objective, and a transparent method for returning information and making true genotoxicity calls. So we explored a number of different data-driven approaches based on that training set that we ran, and ultimately came to combining of several different machine learning types of models to give you that sort of wisdom of the crowd approach across several different applications of machine learning. And then to follow that up and to test the models that we generated, we created some external test sets of new compounds that I will give you an example of in the next couple of slides. What we're looking at here, before we go too much further, I kind of wanted to give you a little bit of a concept of machine learning, because I think this term gets tossed around a lot and people hear about it multiple different times, but some people get kind of intimidated by this and they just kind of ignore it and just say, oh, well, I'll leave that to those computational science people and they'll just figure it out for me. But it's really a fairly simple sort of concept when you think of it just in terms of pattern recognition. So I put an object up here that, and I'm sure we're all very familiar with. This is a stop sign, and I'm sure you recognized it as soon as you saw it come up on your screen. There's a number of different characteristics or patterns within the stop sign that we can extract from that object, and we can sort of look at them in pieces. So we've got the word stop, and then we've got the red color, and then we've also got the shape, that sort of octagonal shape. Those are all different characteristics or factors that go into making up that stop sign. And in fact, they're all the different things that our brain essentially puts together to recognize all those different patterns and ultimately have us make some sort of response to that. We see the stop sign, we are going to stop at that intersection. We can apply this to new situations because of the different patterns that we've understood based on our learning, and we can apply that to new situations and new experiences. So in this case, when I put up an unknown slide that looks very similar to this, so we can see the same color, the same shape, but we don't know the word that is there. In this case, it's also in the context of it's at an intersection, perhaps I'm in a car. I've used my previous learning, my machine learning in this case, that my brain is doing to recognize the different factors and then make a prediction as to what I'm supposed to do here, which would be stop at that intersection, even though I don't speak the language that is on that sign. This is really what pattern recognition is about in terms of machine learning, and that's essentially what I'm asking these different machine learning models to do with the data that we are obtaining from our method. So we were using 3 different models in this case to specifically go into the details. We've got logistic regression, we've got artificial neural network, and then we have a random forest model. We've combined them all together and we take a majority vote of the way that those call, and we get information back on genotoxicity assessment. Now, I don't have the time to go into all the different examples here. I'm just going to show you one specific example here where we're looking at the different compounds. That's our 84 chemical set along the x-axis. So those are all each of the individual chemicals, and then they're broken up into those categories of antigen on the left, clastogen in the middle, and the non-genotoxicant over on the right. And you can see on the y-axis, we're looking at probability of being called a clastogen, and it's pretty obvious from this, those compounds that are in red circles, that's the clastogen group. Those have the highest probability of being a clastogen based on that model call. The antigens are much lower, and for the most part, all the genotoxicants are low as well. So we take basically 80 to 90% predictivity or probability of make these calls, and we call that a clastogen. So we've done that for all the different types of classes of compounds, the antigens and clastogens, and we get an assessment of the performance of those different models. And across all of those different models that we combine together, in terms of a priori clastogen, antigen versus non-genotoxicant classification, we get a performance of 94%. So 94% accuracy in terms of predicting whether a compound was a clastogen, an antigen, or a non-genotoxicant. Now, that's pretty cool when we're looking at a test at a training set, but we already know what that should be. So we need to apply sort of an external test set. So we took 54 additional chemicals that had not been part of the model. We haven't seen the data from this yet. We ran them through our system, and we saw what the predictions would be. And in this case, the established mode of action of those compounds, we got 92% concordance with a priori classification of those compounds. Very good performance in terms of predicting mode of action of genotoxicity. So in terms of conclusions, we have created this add and read multiplexed assay that provides an exceptionally valuable information in terms of genotoxicity potential. Not just telling you whether something is genotoxic or not, but giving you that extra layer of information to tell you whether it's a clastogen, an antigen, or a non-genotoxicant. Those several machine learning approaches were very successfully used and highly performing in terms of their ability to take those multiplex data and turn them into mode of action calls. Similar to what I did before with the microflow assay, we did an interlaboratory performance assessment. There's another paper that's cited there, and you can see the excellent performance that we achieved when we ported this over to other different laboratories in terms of having them run it in their labs and looking at the same compounds that we did. Very good performance that was achieved when we did that interlaboratory trial. So ultimately, the outcome is this high-content method can be used to synergize with the high-throughput, easy-to-use instrumentation, such as you would find in the Miltenyi portfolio. And that brings us to our takeaway here. This combination of high-throughput assays with advanced machines can be exceptionally powerful. And the way that you get that is it really comes down to more information can lead to better decision making. The kinds of decisions that you need to make in terms of your pipeline, whether compounds move forward, whether you keep something back on the shelf or you're actually moving it ahead in your pipeline, this is the kind of information that you need that's much more informative as opposed to the traditional way of getting this where it was just genotoxic or not. In terms of internal decision making and going back and having the chemist reformulate compounds or pharmaceuticals, it's much easier to work with those people if you have more information. If you basically just come back and say, it's genotoxic, fix it please, that's not the best situation for you to work with as opposed to, well, it seems to have some indications of clastogenicity. That gives them a bit more information to work with in terms of redesigning compounds. We're making continued improvements that will hopefully deliver additional advantages to this combination of our methods with those Miltenyi machines, such as the express modes that Jakob was describing earlier. And then finally, in terms of what's happening at Litron, additional new kit formats that we're exploring to look at even deeper levels of information, going beyond mode of action of genotoxicity and actually looking at mechanism. So we can define some of the exact kind of DNA targets that are there as opposed to just looking for, well, this compound is causing DNA double-strand breaks. Well, how is it causing those double-strand breaks? Is it interacting directly with DNA, or is it interacting with some of the enzymes that are important for DNA management? And with that, I will say thank you.